All right, our next estimate, or our next question in the physical biology exam is, how long does it take to exhaust the ATP pool in the absence of ATP synthase, let's say in E. coli? Okay, and so to know this, we need to know two things. First, what's the rate of consumption, which we calculated recently, and also how much ATP do I have? Okay, so if you look at the concentration of metabolites in E. coli, and these are ordered according to from highest concentration to lowest, you see that there's on the order of 10 millimolar of ATP inside the cell. 10 millimolar. Okay, and what does this mean in terms of the number of molecules? Okay, so one interesting exercise for you to do is that 1 micrometer cube, which we said corresponds to 1 femtoliter, it's such that in E. coli, if you have a molecule, one single molecule within this 1 micrometer cube volume, the concentration of that one molecule will be on the order of 1 nanomolar. So basically, if you have one molecule, its concentration in E. coli, it's 1 nanomolar. If you have 10 molecules, its concentration is 10 nanomolars. And this is a super useful number to have for whatever your favorite cell type is. Like, for example, I know that in the fruit fly nucleus, 1 nanomolar corresponds to 100 molecules. Okay, so what does this tell us? This tells us that the number of in E. coli of ATP molecules is then what? Is 10 nanomolar, millimolar, sorry, times 1 molecule per nanomolar. Okay, what's 10 millimolar? 10 millimolar is 10 times 10 to the 6 nanomolars times 1 molecule per nanomolar, which is on the order then of 10 to the 7 ATP molecules. Okay, and what's the rate? So basically the time to exhaust the ATP pool is then what? The number of ATP molecules in E. coli divided by the rate with which E. coli is using up these ATP molecules. And we know this is, on the top we get 10 to the 7 ATPs. And in one of the previous vignettes, we calculated, if I remember correctly, that we're dealing with a few times 10 to the 6 ATP molecules per second. Okay? So basically what you learn is that the time to exhaust the ATP pool is 1 over few times 10 seconds. And remember, 1 over few is like 1 over 3, so it's basically 10 divided by a few is a few. So basically what I'm telling you is that in a few seconds, you exhaust the ATP pool, which is pretty amazing. Like, how this gives you a sense of how quickly the cell is churning ATP to keep up with the metabolic requirements of the cell.